Hello everyone, myself Vani Singh and you all are welcome to my channel. So, you all are requested to like and subscribe my channel and share also. So, today we will be discussing about the replication process. So, before going to the process, you all are requested to watch my previous video about the enzymes. So, let's have a look in the process of the replication. So, I will be discussing points per points. We know there are the 4 nitrogenous bases in the DNA: A, G, T and C. It simply means adenine, guanine, cytosine and the thymine. And in case of RNA, it is the T means thymine. It has been replaced by the uracil. So, let's have a look on the process of replication. So, the first step is the activation of the nucleotide. So, what will be happening here? AMP, CMP, GMP and TMP is the monophosphate and it has been converted into the triphosphate. As you can see in the blackboard, ATP, TTP, CTP and the GTP. TP simply means here the triphosphate. So, in the first step, the monophosphate has been converted into the triphosphate. And one more important thing is here to tell you about the when they are in the AMP, CMP and so on, in the monophosphate condition, they are in the inactive form. So, they will become active only when they are in the form, means triphosphate. So, this one is the inactive form and this one is the active form. My point is getting clear? This one is the inactive form and this one is the active form. So, next step is the opening up of the DNA helix. As we know, the DNA has been spirally coiled. So, to open the DNA helix, we need the enzyme helicase. The helicase is simply opening the DNA helix. And then after opening of the DNA helix, one more enzyme is there, topoisomerase. So, when the two strands has been separated, the tension between the two strands has been created. So, to release the tension, the enzyme which is needed is the topoisomerase. Next enzyme which is helping here is the single strand stabilizing protein. The single strand stabilizing protein is simply just helping in the stable of the DNA. So, I hope this one is clear to you. So, next point is here, recognition of the ori site. Ori sites simply means origin site. So, in prokaryotes, there is only one ori site and in case of eukaryotes, there are many origin sites. So, I hope this one is clear to you. So, next is this is the structure of the DNA when the DNA strand has been released. Okay. So, this structure is called as the replication fork. So, one more thing I want to tell you, that the newly synthesized RNA is from 5th end to the 3rd end. As we know, in DNA there are 2 ends, 3 end and the 5th end. So, this one strand, the strand which is from 3rd end to 5th end is acting as the parent strand. And here you can see one more strand that is from 5th end to the 3rd end. So, these two strands are acting as the parent strand. So, now this parent strands needs to be replicated. So, they will be getting the newly synthesized DNA. It will be from the 5th end to the 3rd end. But this strand is then discontinuous. Why? Because the primase enzyme is involved here. So, when the primase enzyme is involved, the strand will be in the discontinuous form. Now, one more question is there. This one strand from 3rd end to the 5th end is called the leading strand. also known as continuous strand, and the parent strand, which is from the fifth end to the third end, is the lagging strand, also known as the discontinuous strand. So as we know that here the primer enzyme is involved, so the nucleotides are in the discontinuous form. So by this the discontinuous strand name has been given the Okazaki fragments. Okazaki fragments simply means when the strand is not in the continuous form, it is in the discontinuous form. So after all this, when the nucleotides has been added, okay, so after that the proofreading will be there. So in proofreading, two enzymes has been involved. Firstly epsilon, and the second enzyme which is involved is the nuclease enzyme. So these two enzymes will be helping in the proofreading. So after the proofreading, the repair will be there so that they can check any damage or any correction is there or not. So by this the process of replication has been completed. Thank you.